Okay, so now lots of words here. Okay, now we're to the third part of this, so to speak, the electron transport chain. And it's the energy in these electrons which is going to end up in ATP bonds. And again, most of the ATPs by far that come from this respiration breakdown come from this step. This step is also aerobic, like the Krebs cycle was, and like the link reaction, which I kind of forgot to mention back there. Okay, so the electron transfer chain transfers energy in a series of coupled, there's that word again, that means one reaction releases energy, the other stores it. And so we are going to have a gradient formed here that is electrochemical. That means on one side of a membrane, you might have more chemicals, but you also have more charges, right? And in this case, these chemicals are protons, right? Also known as hydrogen ions. So this also occurs in chloroplasts. We'll get back there later. And if you're a prokaryotic cell, right? Let's pretend now this is a prokaryotic cell instead of like a mitochondria. This gradient is just going to occur between the inside of you and the outside world. We have here electrons being delivered by these two, where they pick those electrons up, glycolysis, link reaction, and Krebs cycle, right? And those electrons are going to be passed down this chain till they get to the other reactant here, oxygen, the final terminal electron acceptor. Notice terminal is in there, and so that word might be in a question, and you need to remember that that means final. Okay, so look at this. Aerobic prokaryotes use oxygen as a terminal electron acceptor, while anaerobic ones use other molecules. What this means is maybe we're going to tell you a story about one of those other anaerobic prokaryotes and see if you can apply your general knowledge of how this process works, electron transport chain, to a story where something other than oxygen is at the end, but the same end result is accomplished. Energy from these electrons is going to end up in ATPs. Okay, so this pretty much says what we've said and what we're going to say in a diagram in just a second. So here I just want a couple things here. The electron transport chain doesn't make ATP directly, but as a result of that chain and the gradient, a process called chemiosmosis happens, which makes those ATPs by that kind of phosphorylation called oxidative, right? How do you make an ATP? You add a phosphate to ADP, so all of this is here, right? An electrochemical gradient again. Now they identify that as hydrogen ions, and they expect you to know that that's what protons are, right? The language of the syllabus is the language of the questions. And this membrane, this potential to do work, this gradient, is going to be across the inner mitochondrial membrane. So here we have that inner mitochondrial membrane, right? Here's the matrix deep inside the mitochondria, and here is that intermembrane space. This is where the protons are going to be pumped into, right? And this is where, because they're now more concentrated out here, and this thing allows them a pathway to diffuse back in, this is the chemiosmosis thing. The actual movement of them is the chemiosmosis thing, which very mechanically helps to slap an inorganic phosphate onto an ADP to make ATP. Okay, so this is again known as oxidative phosphorylation. They want that word in your vocabulary. And then we'll get to photosynthesis, because the phosphorylation here is driven by the oxidation of molecules, adding and gaining electrons, and here it's driven by light. So here they throw this in, and I'm not sure why. This is more of a kind of a regulation thing that would be more evolution stuff, but here it is anyway. It's in this unit. If you decouple, if you take this part out of it, the oxidative phosphorylation, from electron transport, in other words, maybe you block these things and the electrons get transported and you don't create a gradient. Or maybe you create a gradient, but you block this thing, that thing called ATP synthase, which I should have mentioned before, right? That's the lollipop structure looking thing. Then you can have all this electron flowing, generating heat, but not making ATPs. So if you're really cold, that might be a good thing to do. If I remember, I'm going to come back to that in one of the very last slides where I have a better diagram to kind of show that idea. Okay, so here's your mitochondria, and now here, another great example of how structure is related to function, maybe the classic one in cell biology, is that this inner membrane is folded. What's that allow? Thousands of copies of what was on the last diagram, just one copy of, so to speak, this electron transport chain and those ATP synthases. If you fold this membrane up, you can have a whole lot more than if it was not folded up and just kind of flat like that. Okay, so here's another example of how structure is related to function. Since that intermembrane space is small, when you pump protons in there, right, it's easy to make a gradient. Proton gradient, not protein. This is a kind of common vocab mistake, so you don't want to say that. We have, I think, everything we've seen here, except again a mention that in prokaryotes, they are going to pump their protons not into a little intermembrane space, but simply out of themselves. And remember their heritage. These mitochondria, this would be the intermembrane, sorry. These mitochondria are just like, that's the intermembrane, they're just like prokaryotes that got captured inside another membrane. So, here we have which stages make ATP, back to this, right? This stage, this stage, this stage, but look how many more, these are maximum amounts, this stage makes. Which of these use oxygen? Not this one, anaerobic. That also is the one that's not in the mitochondria, notice that, right? But the step where pyruvate enters, the step where Krebs cycle happens, and the step where this happens, all of those are aerobic, require oxygen, but not directly, which is where we're going next, right? Here we have the idea that none of these things really need oxygen. What they need is NAD. Glucose to do glycolysis needs NAD. In all those steps of the Krebs cycle where NADH was made, NAD was needed, right? And so if oxygen is low and it can't take electrons from the chain, so NADH can't drop its H off and become NAD again to let these steps happen. That's what really makes these steps come to a halt if oxygen is low. All right. But now here, out in the cytoplasm, again, in the cytoplasm, some cells, vertebrate muscle cells, and some kind of fungi and some other kind of things have evolved these two kinds of fermentation. Now we'll go back to the syllabus, right? They allow glycolysis to proceed. What's the benefit of that? You make these little bit of ATPs for every glucose you break down. It's not much, but it's better than nothing, right? Absence of oxygen here, and it produces organic molecules including alcohol and lactic acid as waste products. What fermentation doesn't produce is ATP. Glycolysis produces ATP, but fermentation allows it to keep happening because here we go, fermentation regenerates NADs from NADH. So that lets the glycolysis keep happening. So this just a quick note. They could tell you this story. Do you know about all this stuff? Even if glucose is not the thing you start with, if we tell you that you can take fats like fatty acids and feed them, chop them up into acetyl-CoAs and start the Krebs cycle, can you predict what would be the difference? Well, we wouldn't make pyruvate, we wouldn't make ATPs from glycolysis, But from that point on, everything else would be the same. That's what would be the same. That's a classic kind of question they like to ask you in terms of, can you tell us what would happen if something changes? Okay, so here the final one is that just like we had in the last thing, a feedback mechanism whereby if this product piles up, it itself can inhibit an enzyme early in the pathway to very efficiently shut the pathway off if we have a lot of its product. Here we have the same thing evolved in a few different ways. I'll just look at one. What's the product that acts as, again, an allosteric inhibitor here? It's ATP itself. If you have a whole bunch of ATPs, this is going to shut off one of the early enzymes in glycolysis to say, you know what, we don't need to break down any more of this. Let's just save it for later because we have plenty of ATPs. So here's back to that heat generating thing, right? If we decouple the electron transport chain from ATP production, right, that can generate heat without making ATPs. What's the problem with making ATPs? ATP shuts this off, and shut this off, you don't generate heat anymore. So that's a neat little evolutionary thing, and it made the syllabus here, and I doubt they'll ask you that, but there you go. So isn't that the way? Boy, this is no joke, but that's kind of a funny picture there. But I hope you're staying distant. I hope you're staying safe. And I'll see you in the next edition, which will be photosynthesis.